all of you, my dear students, in our endeavor of this education through online learning. Student, today's topic or the lesson which we are going to discuss is a part of plant physiology. Plant physiology is a branch of bio which deals with the life functions of the plant. It includes the functioning of cells, tissues, different organs, organ system, and organism as a whole. Today's lesson is deal with some most important fundamental processes like osmosis, which have a very tremendous significance in the life of all organisms. So let's start today's lesson number 4, that is absorption by root. Root not only fix the plant in the soil, giving support, but the most important and life supporting function of the root is to absorb water as well as the mineral nutrients. To understand more about the lesson, we must visit the scope of syllabus. It is there in your textbook. What we are going to talk today or listen today or discuss today are the few points in front of you. Understanding of the various processes which are related with the absorption of water by roots, which includes different types of processes: imbibition, osmosis, diffusion, osmotic pressure, tonicity, plasmolysis, and all. We also discussed the characteristics of root, the structures of full-grown root hairs, general idea of cohesive and adhesive forces, along with the transpirational pool, and close to 4 different experiments which prove that the water is conducted through xylem. Let's begin one by one. Why does plant need water? Plant needs water for photosynthesis because it is one of the important raw material which is used for the synthesis of glucose along with the carbon dioxide. A large quantity of water get evaporated as a water vapor during transpiration for cooling in a hot weather as well as producing a suction force. The water is also required for transportation purpose. Transportation of the substance in a water solution from root to the different parts of the shoot, or from leaf, that is a dissolved food material which is produced by photosynthesis, the different parts of the plant body. Water also provides turgidity, which is very necessary for mechanical stiffness of the plant tissues. Right? So who perform this function of absorption of water? Yes, definitely the root. But what is so special about the root? Why we say that the ability of root to draw water is more than any other part of the plant? Which means the roots are having some characteristic properties which will help to draw the water from the soil. Number one is enormous surface area. Remember, due to the presence of numerous root hairs on the root, the surface area of water absorption increases and therefore more amount of water is absorbed. These root hair contains a maximum concentration of solute inside them. As you can see here, this is the section of root hair, and outside the root hair you will see the water molecules which are accumulated. Please remember that inside the root hair there is a high concentration of cell sap. Cell sap is a substance which is produced along with the water as well as dissolved minerals. So the concentration of dissolved minerals, we can call it as a solute. It is more as compared to the solvent, for example water. And you know very well that the tendency of water is to move from its high concentration, that is from soil, to its low concentration. Correct? So the higher concentration of solute inside the root cell will allow water to move in, or we can say that root will keep the conditions inside their cells so that maximum water is absorbed. That condition is having high concentration of solutes inside the cell sap. Third important property is the thin cell wall of the root hair. Student, this diagram is from our textbook. If you refer your textbook, you will come to know that this is figure 4.2. As far as my suggestion, if you ask me, you have to practice this diagram for sketch and level purpose. If you see in detail or very carefully, you will notice that root hair is nothing but the extension of one of the root cell. So because of the stretching part, the cell wall of the root hair, it will become thinner. It will become thinner. Correct. So this therefore this cell wall will become permeable, and it allows the movement of water molecule and dissolved substances freely in and outer of the cell. Correct. Once the cell wall is also stretched, the cell membrane is also get stretched. It is semi-permeable in nature, which means it will allow water molecule to pass through, but not the larger molecules like dissolved salts. Correct. So the secret of the absorption of water from the soil by root lies mainly of these three characteristics. You have to be very careful, student, because a question can be asked in your exam regarding the structure of root hair. Right? So this one single root hair, it will arise from the epidermal cell of the root, right? Because of the cytoplasmic streaming inside the cell, there is gradually the length of the root hair will increase, and this root hair will have thin cell wall, thin cell membrane. Remember, student, the mechanism of absorbing water as well as the mineral from the soil by the root, it is basically depend on 5 important processes, which we can call them as a 5 main phenomena. Let's start one by one. The first one is imbibition. Before explaining imbibition, let me tell you that you might have seen your mother have soaked some bean seeds or rajma seeds for the preparation of some delicious food item. After soaking it in a water 2 to 3 hours, the size of the rajma beans will start get swelling or increasing. Why it increase? What is the reason behind increase in the size of those seeds? It is because of the imbibition. It is defined as a phenomenon by which living or dead plant cells absorb the water by surface attraction. So what I mean to say, although the water is absorbed, that water will go inside the seed, but seed will not become liquid, or the inner part will not get dissolved in that water. So those water molecules, they are just present on the outer surface of the seed, which will result into increase in the size of the seeds. You also might have experienced this imbibition during the rainy season. If if your home is having any kind of wooden door which are coming in direct contact with the water, so during rainy season they will absorb the water and get swell. Because of that, they will not completely open or close, and hence we will find difficulty to lock them during rain. Second process is diffusion. The definition of diffusion is in front of you on your screen. It is the free movement of a molecule of substance, whether it is solute, solvent, gas or liquid, from a region of their higher concentration to the region of their lower concentration. But the most important thing, they two should be in a direct contact. Example, if you try to drop, or if you release one drop of ink or any dye in a water, the color of the water will change. The color of the water will change. This is due to diffusion. Or if you want to perform this experiment at your school, a beautiful experiment is given in our textbook where you have to add some potassium permanganate crystals in a water in one corner, correct? So, and when you start stirring that water, the color of the water will change, which is nothing but a diffusion. Second important phenomenon or process is osmosis. It is defined as the diffusion of water molecule from the region of high concentration to their low concentration through semi-permeable membrane. Let's understand this process with the help of the picture right in front of you. In this picture, if you see, there is a beaker divided into left side and right side with the help of a semi-permeable membrane. We can use cellophane paper or parchment paper or whisking bag, gourd bladder or egg membrane, which can serve as the semi-permeable property. Left side of the semi-permeable membrane will have a solution with low solute concentration. It means if solute is less, then definitely the solvent is more. So water is more than any solute. But the right hand side of the beaker will have high solute concentration solution, which means in this solution, the quantity of water is less than the solute, correct? So as per the definition, the water will start moving from low concentration of solute to high concentration of solute, or simply it will move from high concentration of itself, that is high concentration of water, to the low concentration of water. यहाँ पे अगर आप लोग देखोगे तो there are hardly 6 solute particles you can see, but here you will see there are more solute particles, correct, which are indicated by the red color bigger circles, and the water molecules are represented by the blue dots, correct. So therefore after some time you will see the solution level on the right hand side of the beaker, it has risen. Let's demonstrate the osmosis with the help of a simple experiment. In this experiment, we will require a thistle funnel, correct? We will cover the mouth of the thistle funnel with a cellophane paper. We will fill it with a sugar solution. We also required a beaker containing a plain water. We will place the thistle funnel in inverted manner and suspend it with the help of a wooden stand. Later on, we have to mark the initial level of the sugar solution on the stem of the thistle funnel, and after a few hours, we can observe that the level of the sugar solution will increase. Now, what is the reason behind the increase in the sugar solution level? Number one, the cellophane paper act as a semi-permeable membrane which allows water molecule to move inside and do not allow sugar molecule to come outside, right? You can modify this experiment, correct? You can modify this experiment by just keeping, instead of water in the beaker, we will take a very dilute or less diluted or less concentrated sugar solution as compared to the more concentration of sugar solution in the thistle funnel. Okay, one more twist we can do, and we can record the observation. Instead of cellophane paper, if we put a rubber sheet and a muslin cloth, then you know what will happen? The rubber sheet, it is completely impermeable. It will not allow water molecule to go inside. The level will not increase. If we attach or if we tie the mouth of thistle funnel with the help of muslin cloth, the pores of the muslin cloth are so large that they would not hold back even the sugar molecules, and the entire sugar molecule or the sugar solution would flow down to the common level due to the gravity in the beaker. Okay, so the two important points from this experiment, what we have understand is that there are two liquids of a different concentration, correct? Now if you see in this picture, the sugar solution is having more amount of a sugar molecule but less amount of a water. If you see in the water, there is more amount of water but less amount of the sugar. And it is very important to understand that concentration can also be visualized from the side of the solvent as well. And that is why there are two different regions. The region one is more solute, less solvent, and region two is more solvent, less solute. And this is nothing but this different concentration is nothing but like a tonicity. Correct? So how long this osmosis will continue? As I say that water will move inside at initial stage. But theoretically, if we see that osmosis should continue until the concentration of water molecule becomes equal, outside is equal to inside, correct? But it will not happen. It will not happen. The column of the sugar solution, which is rising in the thistle funnel, it will keep on rising, correct? And the height and the weight of this solution would also increase. Such increase in the height and the weight of the rising column, it will reduce the osmosis. What I mean to say, if the water continuously flow inside, slowly the water level will increase. But after some time, because of the gravity, there must be some kind of pressure is generated over here. And that pressure will not allow the next water molecule to come inside. And student, there are two types of osmosis, endosmosis and exosmosis. Endosmosis is defined as the inflow of water into the cell. If you keep the resins in a water, it will get swell. That is called as endosmosis because water will move from beaker or bowl to the resins. Exosmosis is defined as the movement of water molecule from cell to the outside. For this, you have to keep the grapes into a a salt solution. Correct? After some hours, after few hours, you can see that the water from the grapes has come out and the grapes will get shrink. Osmotic pressure. Osmotic pressure of a solution is a measure of its tendency to take in water by osmosis. Now student, it is a similar experiment which we have just discussed. Correct? Same thistle funnel with a concentrated sugar solution. The cellophane paper is tied on the surface, invertedly suspended into a beaker containing water. And after some time, you will see that water level has increased. But we will do some twist over here. Now, in this setup, we will immerse or we will insert a piston. That piston will have certain amount of load. Correct? Okay. And if you see that because of the keeping this load, the piston is creating a pressure on the sugar solution. Correct? And that pressure is not allowing water from beaker to come inside. And hence there is no rise in the water level inside the thistle funnel. So if you try, correct, if you try to remove the load or the weight from the piston, it might happen that some amount of the piston will be rising or it will be pushed upward because of incoming water. So therefore we can say that osmotic pressure of a solution, it is a measure of its tendency to take in water. If you increase the pressure, water will not come inside. That means there will not be endosmosis. If you release the pressure, the water will come inside. Next concept is tonicity. Tonicity is defined as a relative concentration of a solution that determine the direction and the extent of diffusion. Correct? Now what I mean to say that when you look at the earlier slides, what we have just discussed, right? So whether the water will come inside or water will go outside from the semi-permeable membrane, that is called as the direction. And extent means what? How much time it will come inside or it will go outside, that is called as tonicity. Now it is totally depends, student, on what is the solute and solvent concentration across the membrane, across the semi-permeable membrane. Based on this, we are having three types of conditions: isotonic condition, hypotonic condition, and hypertonic condition. Let's see one by one. Isotonic condition. The relative concentration of water molecule and the solute molecule on either side of the cell membrane is same. In such solution, there is no movement of water molecule across the cell membrane. What I mean to say, student, this is a plant cell and this is the animal cell. Correct? Now, if you see one water molecule is coming in, the one is going out. So it means inside the cell and outside the cell, there is no change in the number of water molecules or even in the concentration of solute. So hence the shape of the cell, second type of the condition or tonicity is known as hypertonic condition. Now what happened in hypertonic condition? The solution outside the cell, it will have higher concentration of solute, as compared to the fluid inside the cell. Correct? It means outside the cell there is more solute but less solvent. But inside the cell we will have more amount of solvent, less amount the solute. So water will move from inside of the cell to outside of the cell. And therefore we can say that here the exoosmosis will take place. Okay? Here the exoosmosis will be take place and cell, it will get shrink. If you see in case of this RBC, that is animal cell, the size has significantly reduced. Third condition is the hypotonic condition or hypotonic solution, in which what happened? The solution outside the cell, it will have the lower concentration of the solute as compared to the fluid inside the cell. It means सेल के अंदर बहुत ज्यादा सोल्यूट है और सेल के बाहर बहुत कम सोल्यूट है. When I say that outside the cell there is a less solute concentration, then whose concentration is more? The water concentration is more. Inside the cell, solute is more, water is less. That is why water will move from its high concentration to low concentration through semi-permeable membrane. And because of this inward movement of water, we can say endoosmosis will take place. Endoosmosis. Correct? And the cell, it will become turgid or it will get swell up. Animal cells, since they do not have any kind of protective membrane like cell wall, a rigid structure in case of plant cell, animal cell may burst if we keep it in hypotonic condition for a longer time. The next concept or the next important process which will favor the absorption of water and minerals by the root is active transport and passive transport. Definition: active transport is the passage of the salt or the ion from its lower concentration to the higher concentration. Imagine, from its lower concentration to higher concentration through a living cell using energy from the cell itself. This is what is the picture. You can look at it. This is semi-permeable membrane. Left hand side, the blue color crystals or the square shaped structure, they represent ions or the salt. The concentration is very less on the left hand side, but on the right hand side, the concentration is more. Still, these ions will move from left to right, that is from lower concentration to higher concentration, but ATP will play an important role in the absorption of these ions. Exactly opposite is the passive transport. It is very much similar to that of diffusion. The passive word refers that there is no requirement of any energy and there is a free movement of molecule from their high concentration to their low concentration. Correct? Student, remember that active transport, it will be required for the absorption of certain nutrients like ion of nitrates, sulfate, potassium, zinc and manganese. Because their size is so much high that it won't be absorbed directly into the cell. So that's why we will require energy. Student, every living plant cell, directly or indirectly, is in contact with fluids. That is the root hairs. In particular, they are surrounded by the soil water. All such cells, they are subjected to the osmosis. And water will continue to enter inside the cell or inside the root cell as long as the cell sap is having more concentration of solute than the surrounding soil. But a time will come when a cell will reach to a state where it cannot accommodate any more water. That is, the cell will become fully distended. As you can see in this picture, the yellow color structure is a vacuole and there is a continued inflow of water causing maximum stretching of the vacuole resulting into the stretching of the cell. Such cell is called as turgid cell and the condition is called as turgidity. Now what is happening over here, if you see the vacuoles, they are swell up and they are pushing the cell wall towards the outside. It means the cell wall will get stretched. And why it happens? Because in this pressure is known as Turgor pressure. The Turgor pressure is the pressure of the cell content on the cell wall. If you see, these are the number of plant cells and this blue color structure, it is the vacuole full of with the water. It is getting stretched and it is pushing the cell content towards the periphery. But the periphery is having cell wall which is very rigid structure. Correct? So it will press, that vacuole is pressing the cell content towards the cell wall. Correct? So we can say that the pressure exerted by the cell content on the cell wall from the within the side of the plant cell, it is called as Chugger pressure. Correct? But the pressure of the cell wall, correct? The pressure of the cell wall is also acting. It means equal amount of pressure it will be exerted by the wall on the cell content, that is called as wall pressure. Remember, if at any given time cell wall is unable to bear the Turgor pressure, it will rupture and the cell content, it will burst out. This will exactly happen when certain fruits and vegetables sometimes burst naturally on the plant. When turgid, the cell is somewhat in balanced state. Always remember, no more water is entering and leaving it. It means Turgor pressure, it will counterbalance the wall pressure as well as. Correct? So therefore, there is no further absorption of water. Student, when a living cell, for example, the cell of the leaf of an aquatic plant, when we place it in a fresh water, it will remains in a fully distended condition. That is, the plasma membrane will remain in close contact with the cell wall and it will be pressed against the cell wall. Correct? If this plant cell, if you take it out from the water and if you keep it in a 5% salt solution for some minute, it will lose its distended appearance because of plasmolysis. What is plasmolysis? It is the shrinkage of the cytoplasm from the cell wall, and due to this, the cell will go in a state. That state is called as flaccid state, and the condition is called as flaccidity. Now, as you can see here, this is the plant cell, correct? When you keep the plant cell in hypertonic condition, if you remember, hypertonic means when outside the cell, the concentration of solute is more. I just said that we have to keep that the leaf of the aquatic plant into 5% salt solution, into the 5% salt solution. So what will happen? Water from the cell, it will come out, that is lysis of plasma, that is liquid state. The cytoplasm, the water from the cytoplasm, it will come out and cell will get shrink, or it will become flaccid. Condition is called as flaccidity. Definition: flaccidity is a condition in which the cell content is shrunken and cell is no the stages of plasmolysis. If you see here, this is the normal cell. This is the normal cell where the cell membrane is close in contact with the cell wall, but slowly, the cell membrane had started losing its contact with the cell wall, and it is shrinking, or it is moving towards the center. Now in the C diagram, if you see, from all the side, the cell membrane has detached, and finally all the cell content, it will get accumulated at one stage or one corner. What is the reverse of plasmolysis? If due to plasmolysis, as you can see in this picture also, the water is going outside and cell is going to shrink. But the recovery or the reversal of the plasmolysis, it is known as deplasmolysis, as you can see here. As you can see here, the water is going in. Again, this vacuole will store the water and it will bring the cytoplasm and the cell membrane and entire cell coming back to its original shape. This is called as deplasmolysis. Is this plasmolysis is really helpful? There are certain application of plasmolysis, student, even we can experience in day-to-day life. Number one, salting of meat. While cooking the meat, we used to marinate the meat with the help of certain spices as well as salt. So because of adding of all this thing, the water will be drawn from the muscle cells, correct? And it will make the meat soft and juicy. Adding of extra salt in pickle is again a method of killing the bacteria by plasmolysis, because that extra salt present in the pickle, and when bacteria start attacking or start growing on that pickle, it will draw the water from bacterial cell. Even farmers, they use certain sprinkling of the salt to kill the weeds, because this the salt which is sprinkled around the bases of the weeds will draw the water from the root cells, and the root cells will die. Similar thing will also happen if there is an excessive application of fertilizers occur, because the fertilizers are also salt. They are the salts of nitrates or carbonates or simply urea. They are going to draw the water from the roots, and definitely it will damage the roots and diminish the entire yield also. Sometimes even the elder people from our family also advise us to gargle with the salty or lukewarm water when we are having a complaint of sore throat. It might be due to certain kind of bacterial infection. So when we gargle with the salt water, that salt water will try to pull out the water from bacteria causing infection, and we may get relief. There are certain uses of the turgidity to the plants. If you can see, the first one is to provide rigidity to the soft tissues. Turgidity, it is providing rigidity to the soft tissues such as leaf. When there are not enough water in the leaf, it will get wilt, as you can see in this GIF. The petiole and the lamina will become loose and the leaf will droop down. Wilting of this leaf is usually noticed when a plant is exposed to hot afternoon sun, when the amount of water lost by transpiration is more. But in the evening, when the transpiration is reduced, the quantity of water is absorbed exceeds the water loss through transpiration, and turgidity of the leaf cell will be restored. Or simply, when you pour some amount of water to the plant, the turgidity will be restored. Second, the turgor pressure helps to push through the hard surface. As in case of mushroom or in case of seedling, as you can see in the GIF, sometimes the root of the certain trees have been seen to crack the walls of the concrete floor of an adjoining building. Even this is again due to Chugger pressure. The root cell, the turgor in the root cell, it will build up the root pressure. If you can cut a well-watered pot plant, for example balsam, few centimeter above the soil, and immediately fix a glass tubing to its mean of a rubber connection, water will start coming out of the cut end of the stem and rise up in the glass tube. This rising of the water can raise the mercury level, in the connected manometer. This manometer is an instrument to measure the pressure. So this upward flow of water is due to heavy pressure from the roots, which is called as root pressure. And because of this root pressure, the water can rise the tube or in the stem in case of natural condition up to the certain height. And the loss of water through the cut stem, it's called as bleeding, which we will discuss more. The next use, that turgor pressure, it will help in opening and the closing of stomata. The opening and the closing of the stomata is depend on the turgidity of the guard cell. Here, the stomata is open because the guard cell is fully turgid, and here the stomata is closed because the guard cell has lost its turgidity and has become flaccid. Guard cells contain chloroplast. Okay, guard cells contain chloroplast. As a result of this, there is a synthesis of glucose during photosynthesis, and also some other chemical changes. The osmotic pressure of the content of the guard cell will increase, and they absorb more water from the outside cell, correct, and thus becoming turgid, correct. On arc outwards, and the aperture becomes widened and thereby opening the stomata. But at night, what happens? In the leaf, the guard cells turns flaccid and their inner rigid wall becomes straight, and the closing of stomatal aperture. Chugger movement, the rapid drooping of the leaf of the sensitive plant like Mimosa pudica, okay, on outstanding as an outstanding example of the Chugger movement. If one of the leaf is touched even lightly, the leaflets will fold within 2-3 seconds. The entire leaf will droop down, and if the leaf is touched somewhat strongly, the wave of the folding and the drooping will spread stimulated leaf to all neighboring leaves, as you can see in this GIF. Slowly, the leaf will recover again and stand erect because the turgor pressure of the leaf will decrease when we are touching to the leaf, right? In this plant, the stimulus of the touch, it will lead to the loss of turgor at the base, yahan pe, at the base of the leaflet. And at the base of the leaflet, there is a structure called as pulvinus. It is given in our textbook. The figure is given in the textbook. Okay. Somewhat such kind of movement even we can observe in insectivorous plants, correct, whose leaves are whose leaves, it will get close when they entrap the living prey. Bending movement of certain flowers, that is sunflower towards the sun, and the sleep movement of the leaf during certain in case of certain plants at night is also due to Turgidity movements. Please remember. Next is the root pressure. Root pressure is defined as the pressure developed in the roots due to the continued inward movement of water through cell to cell osmosis. As you can see here, this part is a root hair which is surrounded with the soil water. The water, when it start entering into the root hair and associated with the root cell, the cell will get distended or it will get turgid. And because of that turgor pressure, okay, created over here, and lesser of the wall pressure, the water will move from cell A to B, from B to C, from C to D, E, F, G, and later on it will reach to the xylem. This movement is called as cell to cell osmosis, and this will happen, okay, this will happen because of the root pressure. In our textbook on page number 47, you will see such a diagram. That diagram is very important as far as your examinations are concerned. Please remember all the labels which are given in the diagram. Next is guttation. It is defined as the loss of water in the form of water droplets. And mostly it is found in the plants like tomato, grass, banana, or some species of ferns. When the root pressure is high enough to force the water all the way through the stem, and that water will come out through the end of the leaf veins. And this water, it will appear in the form of small tiny droplets. And they are always located on the margin. Please remember, it will not come out from anywhere. It will come out only from. Now, certain important experiment to prove that the root absorb water. We have to take two test tube containing water along with the oil. One test tube will have a plant like a balsam which can show the absorption of water. The another test tube will not have any kind of plant. After a few hours, we will see that water level in the test tube A, it has decreased. It is due to the water will get absorbed by the plant. Experiment. Second experiment is to show that water is conducted upward through the xylem. A medium-sized young balsam plant we have to take, wash and place it in a beaker which contains stain like evosin. The root should be completely submerged in the solution, and this setup is kept aside for 3 to 4 hours. Later on, when you take the TS of root, TS of stem, and TS of leaf, and when you examine under it microscope, the xylem vessels will appear red in color. Correct? Because these will be stained due to the dye which was used, that is evosin. Experiment number 3, to show that conduction of water will take place through xylem. We have to take two leafy shoot for those of the balsam plant again, which have been cut under the water to prevent any air bubbles getting into it. Keep their lower ends dipping in the water for some time. Then remove about 3 centimeters of the outer portion, that is phloem of the stem in one of them, as you can see in this setup. Now here we have to take care that the internal part should be intact. It should be intact. And in other beaker, we have to remove the equal part of, or equal centimeter, that is 3 centimeter part of the middle area, from the middle area, that is xylem. We will not disturb the outer portion in the stem from beaker B. The shoots are then fixed to the stand, and they are allowed to remain for about 2 days with their lower end immersed into the water. It will be found that the leaves of the first week remain turgid and stand out almost normally. But those in the second beaker, the second twig, these leaves will get wilted and it will droop down. This will prove that water is conducted upward by the xylem, because we have damage over here the xylem. That's why the water will not reach to the leaves. Experiment number 4, to show that the food from the leaves is conducted downward through phloem in the stem. Cut the ring around the stem of a healthy potted plant or around the thin twig of guava, or for that matter any tree. It should be deep enough to penetrate the phloem and cambium, but not the xylem, which is a place at the center. This process, student, removal of the phloem is called as girdling. It will be seen that the cell sap, it will start oozing out from this side. Okay. Further cut the margin of the stem, showing thereby that sap is in the peripheral parts. Now, after some time, you will see that the swelling will occur on the upper part. This is due to the accumulation of food. The part which is present below this cut region, it will stop growing and it will die soon. Correct? So therefore, we can prove that the food is transported or conducted downward through phloem, because we have cut the phloem here. Student, there are certain forces who actually contribute the ascent of sap apart from uh, uh, the one which you know, that is root pressure and transpiration pool. One of the important forces is capillarity and second is adhesion. Let's discuss one by one. You know about the root pressure. We have already discussed. Capillarity, a narrow diameter of a xylem vessel, it will cause the water from lower level to rise up to fill the vacuum which is created by the leaf due to loss of water in the form of transpiration. In other words, if I would like to explain what is capillarity, I will request you, when you go to your school, go to the chemistry lab and ask that teacher to show you the capillary. Capillary tube is a glass tube which is open from both the ends, which is having a diameter very small, and when you keep it in any kind of fluid, certain amount of that fluid will rise in the capillary tube. Problem will be that capillary tube is transparent, so you have to take any colored solution. Or simply I will give you another example of capillary. Correct? Whenever we are consuming a cold drink with the help of straw, when we immerse the straw inside the cold drink, certain amount of cold drink rise up in the straw. Does this happen? Why does this happen? Because that straw, now it serves as a capillary. Because of certain force or certain pressure of the cold drink particles, it will try to rise up to the certain level. This is called as capillary. And that rising of the cold drink inside the straw, you can correlate with the rising of water inside the xylem. Again, this is also caused due to capillary force. This is also caused due to capillary force. The third force which act on the ascent of sap is transpiration pool. As the water is lost from the leaf due to transpiration, more water molecules they are pulled due to the tendency of water molecule to remain joined. As you can see in this GIF, two water molecules they are moving from the root, entering into the xylem, reaching to the stem, and from the stem they will go to the leaves, and after leaves they are getting evaporated. So one water molecule pulling the other water molecule is called as cohesion. As you can see, there is a similar attraction between similar molecules. We called it as cohesion. Correct? And not only the water is rising, it is also rising the minerals or the nutrient or the salt or ions. This is due to adhesive force or adhesion. It causes water to stick to the surface of the cell, thus drawing more water molecule from below when the leaf cells loses the water. Correct? And along with that water molecule, the ions like zinc, sulfur, or phosphorus, potassium, sodium, nitrogen will be absorbed.